everyone. Welcome to Rbiochem.matrix, Biochemistry and Biotech Hub. Today we are going to discuss about the biochemical test. Part 1. What are biochemical tests? Instead of reading these lines, I will explain you what is biochemical test. A biochemical test means, for example, a patient is coming to a doctor by saying certain symptoms. He is having stomach ache, he is having diarrhea, vomiting, etc. etc. So, do you think the doctor will be prescribing the medicines immediately after hearing all the symptoms? Definitely not. What he will be doing, instead of giving all the medicines directly, he will be asked the patient to take several tests. He will be sent to a laboratory to take test. What are those tests? Can be a blood test or a urine test or a fecal test, whatever the test it can be. What a microbiologist will be doing to give a report, first step he would be doing is the staining. What type of staining he will be doing? He will be doing gram staining as the preliminary test. First, he will stain the bacteria. He will identify whether the bacteria is gram positive or negative. Then he will be doing this biochemical test. Certain bacteria will react to certain type of biochemical compounds. What is this biochemical compound? Biochemical compound is a compound which is present in certain bacteria. It can be an enzyme or it can be any compound which shows some kind of reactions. Okay, so it is a chemical compound which is present in a living organism. That is known as the biochemical compound. Why we are doing this test? We are doing this test for screening, prognosis and therapy monitoring in the diagnosis and treatment processes. So after identifying a bacteria reacts with these types of biochemical compounds, the microbiologist will come to a conclusion that this bacteria will be this species. So after confirming that, he will be doing the AST. What is AST? AST is the antimicrobial susceptibility test. What this test is done for means, so this test is done to identify whether the bacteria is susceptible to certain antibiotics or whether it is resistant to those antibiotics. He will be giving a complete list as the report to a doctor. The doctor will go through those reports and then only he will start to prescribe a medicine for the patients. What are the different types of tests we have? We have indole test, methyl red test, catalase test, oxidase test, triple sugar iron test, motility test, mannitol, citrate, urease, coagulase and Voges-Prosker test. Instead of doing all these 11 tests, the microbiologist will do only 4 tests, and that test is going to be called as the IMViC test. As I have told you earlier, it is the combination of 4 tests: indole, methyl red, Voges-Prosker and citrate test. The small I is used generally for the pronunciation purpose. And now coming to the first test, what is this indole test? Certain bacteria will be producing tryptophanase enzyme. This tryptophanase enzyme will break the tryptophan and amino acid into indole, pyruvate and ammonia. We will be adding a reagent known as the COVAX reagent to identify whether this bacteria is doing this process or not. If the bacteria is showing the positive result means it is degrading tryptophan and forming the compounds, which is indole, pyruvate and ammonia. Then this reagent will give a color change, and it will be giving a red color ring at the top of the liquid. What are the procedure which we are going to follow? Remember, whenever you are performing a biochemical test, after doing the gram staining, we have to do we have to prepare certain medium for that. The medium will be the agar medium, or it can be a broth. What is the difference between an agar and a broth? Agar is something which is a solid type, whereas the broth will be in the liquid form. Now, after preparing the broth, we are going to do inoculation. What is an inoculum? Inoculum is something where we are going to take the old species and we are going to add it to the new area. And after adding this, we are going to grow them. That is known as the incubation. We are going to develop the bacteria in number. And after this incubation, we will be adding, whether we can add reagent or we will be observing the results directly. So here for this step we are taking tryptone broth, tryptone water, and then we are adding this species. It can be any species. For example, we are taking E. coli. We will be adding this E. coli into this. And what is this type of needle known as? This is known as the inoculation loop. Okay, and here the part which you can see can be made up of nichrome. Okay, first we will be heating this needle for sterilization, and then only we will be taking this species, and then we will be doing inoculation. After inoculation, we are going to do incubate. We are going to incubate the tube. For incubation, we are going to do at 37 degrees Celsius for 24 to 48 hours. After 24 hours or 48 hours, we are going to take the tube and we are adding the Covax reagent. After adding this Covax reagent, we will be observing the result. If it is going to be a positive result, there will be a formation of red ring. If it is a negative result, there is no color change, it will remain yellow itself. Now tell me one thing, what will be the color of this Covax reagent? Please do comment. Now we will be identifying what are the species which shows positive indole test and what are the species which will show negative indole test. The positive indole test are shown from this E. coli, Proteus vulgaris and some common Klebsiella species. And from the negative thing, it is Enterobacteria, Erigonines, and then Klebsiella pneumonia and Proteus mirabilis. And these are the some common application for the differentiation of the Enterobacteriaceae family, and then for identification, and then for quality control, and then used for the taxonomy purpose also. What is Covax reagent? What are its composition? The main three composition are para-dimethylaminobenzyaldehyde, isoamyl alcohol or the N-butanol, and then a third is the acid, which is the concentrated HCl. What this DMAB does is, it will be forming a colorful complex when the tryptophanase is breaking down, and when this reagent is combined with this indole, it will form the colorful complex. And then this third one is going to change the pH. Okay, it will be changing the pH, isoamyl alcohol, and the third one is just acts as the acid catalyst for the process. Okay. Now coming to the methyl red test. What happens in this methyl red test is, the certain bacteria will produce acid from the glucose fermentation. That is the normal principle of this test. So we know, when a product, for example, when a substrate, glucose, lactose or sucrose, whatever the carbohydrate product we have, when a glucose is getting fermented, obviously we know an acid has to be produced, which means the pH is going to be decreased. Okay, from the normal pH, the pH is going to be decreased. So if the pH is below than 4.4, it is going to give a red, which is a positive test. If it is going to be greater than 6.8, which is going to give yellow, which means a negative test. So we will be adding methyl red as an indicator. So when what this indicator will be doing, if more acid is produced, it will be giving us a red color. If there is no acid produced, if the pH is still in the basic pH, then we will be getting a negative pH, negative result, which means yellow color. So what is the procedure? We are going to take this MRVP broth. Here also we are using the broth only. We are going to inoculate the species and we are doing incubation. We are going to see the temperature. It is going to be 37 to 35 to 37 degree for 48 hours. Up to 5 days we can keep it for the stable acid production. So then we will be adding this methyl red indicator. After adding this methyl red indicator, we can see the color change immediately. Red color means positive, yellow color or no color change means it is going to be negative. So here the color change for the yellow, the reagent's color is going to be remain. Okay. So what are the positive species which are grown? E. coli and Proteus vulgaris, and here also Klebsiella pneumonia, Proteus mirabilis, Enterobacteria aerogens. So here also some common applications are being given. Now coming to the Voges-Prosker biochemical test. What happens in this Voges-Prosker test? So here also same thing is going to be done. But here, what is the difference between this methyl red and this is going to be a light change. In this test, we are going to check the ability of the bacteria to produce acetyl methyl carbinol, which is the acetone, and non-acidic neutral end product of the glucose fermentation. But there we will be seeing the formation of a carboxylic acid. Okay, so see, glucose is there, the pathway will be formed and then we will be having this acetone molecule is formed. We will be adding the reagents VP1 and VP2. Here we will be adding two reagents. When this is being done, we will be again getting a red or pink color complex. Okay. In VP2, it's going to be KOH. VP2 is KOH, 40% KOH, and VP1 is going to be alpha naphthalol. What is the procedure? We will be again taking the MRVP broth, we are inoculating and we are incubating them at 37 degrees Celsius for 24 to 48 hours and we are adding the reagents. Only one reagent is given. VP2 is 40% KOH. And then if it is the positive thing, we will be having this red color. If not, it is going to be the yellow color. Some species which are grown is Klebsiella pneumonia. Klebsiella pneumonia, for the past two tests we have seen in the negative. But now we are going to see them in positive, and Enterobacteria aeruginosus. Whatever the species which we have seen in the positive for the indole and methyl red, now it is coming in the positive thing. Whatever was positive in the past two tests, now it have went to negative. Okay, like see E. coli, Proteus mirabilis, Salmonella species and all. And some kind of applications has been given for the differentiation of the Enterobacteriaceae family, and then identification and then quality control and taxonomy has been given. Now is this two reagent. What are those two reagent which we have seen? That is VP reagent A, or known as the Barrett's reagent also. Barrett's reagent A is nothing but the alpha naphthol, which acts as the color intensifier. And Barrett's reagent B is the 40% KOH, which oxidizes the acetone into diacetyl compound. Okay, so this one you can see in the previous slide for the reaction thing. Now is the last test, which is the citrate utilization test. What happens here is, this citrate is there. What is the main function of this citrate? Some bacteria can use citrate to grow in the absence of another carbon source. For an organism to be grown, it needs a carbon source. Generally, the carbon source will be either sucrose or glucose. But certain bacteria can use citrate as their carbon source to grow. And they will be producing an enzyme like citrase or citrate permease to transport citrate into the cells. In the citrate test, organisms are cultured on the Simonsen citrate agar, which contains sodium citrate, some new micronutrients and a pH indicator. Okay, organisms which utilize this citrate will also break down the nitrogen source, giving off a basic product, turning the pH indicator from green to blue. Okay, so see here, when citrate is being utilized, it gives the, in the presence of citrase enzyme, oxaloacetate and acetate is given. Again the reaction with the citrate, it will be giving you pyruvate and carbon dioxide. This can go in the alkaline, it can form alkaline products also. What happens if the alkaline products are formed? The pH will be getting raised. When pH is getting raised, the solution will be turned from green to the blue. If positive test, it will form a dark blue color. If it is a negative test, it will remain in green color. Okay, same thing, we will be inoculating them in a slant. Now from the broth we have moved into the agar. What is the agar which is used? Which we use is the Simonsen citrate agar, which contains sodium citrate, which acts as the carbon source, and ammonium salt is being there for what? For the nitrogen source. And then we have the pH indicator as bromothymol blue. And then we have the phosphate buffer to resist the pH changes. And then we are adding this, we have prepared the Simonsen agar and we have tilted it slightly to form a slant. So this is known as the slant and this is known as the butt. This one is very important for studying the triple sugar iron. So we are going to inoculate in the butt and then slant, and then doing this one, we will be incubating it for 35 to 37 degrees Celsius for 24 to 48 hours, or up to 7 days also you can keep it. After this we are going to check for the color change. What happens if the pH has been raised, alkaline products are formed, then it is going to be in blue color, or else it will be in the negative color. Okay, for positive examples, it will be Enterococcus and Salmonella. If it is in green color, which means there is no utilization of citrate and there is no alkaline products has been formed. This is what the result interpretation. Again, there are applications such as differentiation, identification and quality control and all. Stay tuned for the part 2 video where we will be discussing about the urease test and the other tests and all. Okay, stay tuned, and whatever the things which I have asked you for the comments, please do comment, and if you have any doubts, feel free to ask in the comments and I will try to respond in the comment. Thank you.